With RNA processing, now that we have our piece of messenger RNA, our piece of messenger RNA, immediately after we make it, it has a lot of information inside of it, but it's been made within the nucleus. And the nucleus is a really nice, safe place. Everything is safe and quiet and sound inside of the nucleus. But when we send it out into the cytoplasm to participate in translation, the cytoplasm is full of all sorts of dangerous stuff. There's free radicals out there, there's enzymes out there, there could be viral DNA, there's other organelles, there's all sorts of reactions that are happening everywhere, and it's a really busy place. And so the messenger RNA needs to be prepared in order to go out into the cytoplasm. So when it's first made, it is in the nucleus, but it is eventually going to pass out of the nucleus into the cytoplasm. And so there are three things that we do to the messenger RNA to prepare it to leave the nucleus. And we're going to go in a particular order. I don't know if it really matters what order you go in, but you do need to know what those three things are. And so the three things we're going to do are called splicing. Splicing is number one. The second thing that we're going to do is we're going to add a 5' cap. And then finally, we're going to add a poly-A tail. And so these are the three things we're going to do to the piece of messenger RNA, and I'm going to talk about what they all are, but they all have very specific roles. We're going to do something to change the RNA, and you need to know what that change is, but you also need to know why we're doing it. So the first thing is splicing. And splicing involves removing unneeded sections of the messenger RNA. Some pieces of the messenger RNA, which are copied directly from the piece of DNA, we don't actually need to be in the mature transcript that goes out into the cytoplasm. And there's a number of reasons for that, which I'll talk about in class. But we have these two broad categories for what we call these pieces that we are either going to keep or that we're going to reject. And those are introns and exons. It's always seemed to me like exons should be the thing that you're losing, but this is in fact the thing that you're going to keep. And so exons are the information that you are going to keep within that piece of messenger RNA. And the introns are the information that you are going to cut out and remove from the piece of messenger RNA. And so we're going to start out with a piece of messenger RNA. And this piece of messenger RNA is going to have parts that are introns and parts that are exons. So let's call this will be like, this will be intron number 1, intron number 2, and then intron 3. And then we're going to have exon 1 and exon 2. And so what's going to happen is an enzyme is going to come up to this piece of messenger RNA, and it is going to bend it into what we call a hairpin. And what that looks like is you got your piece of messenger RNA, and we bend it around in a loop like this. And when we bend it around in a loop like that, there are 2 points of the messenger RNA that are going to be like touching. And at that area where they're touching is going to be an enzyme. And this enzyme is called a spliceosome. And the spliceosome is going to cut the little piece of messenger RNA at this point. This is going to leave, and then these things are going to be joined together. And this piece that's been bent into a loop, we call this a hairpin. So the spliceosome comes up to the piece of messenger RNA, grabs it by the intron, causes the intron to turn into a hairpin, and then will join the exons on either side of that intron together. And so in this particular picture, the thing that we're turning into the hairpin, this would be the intron, and then these parts that we have on either side, these are all exons. And so let's imagine this is intron 2, this would be exon 1, this would be exon 2, this would be intron 2. And so when we're done, exon 1 will be joined to exon number 2. So when we're done with splicing, our piece of messenger RNA is going to be comprised of nothing but exons. And so it would look like that, and it would look like that. And we do splicing for one of 2 reasons. The first is that, because you could say, well, why do we even have these introns inside of there if we don't actually need them inside of the protein? Why don't they just become removed from the piece of DNA? And there's 2 reasons for that. The first is that much of this is very ancient information. And so you have some protein. Let's imagine it's like ATP synthase. That protein is literally billions of years old. And so it has probably got some information in there that is just old and really not necessary. And so we just kind of cut it out and get rid of it. The other reason is for this thing called alternative splicing. And with alternative splicing, that means that you're going to have one gene, but you're going to splice it in a number of different ways, and you can get many different proteins. So this would be like protein number 1, protein number 2, and protein number 3. That means you have some gene for like how to make muscle protein, myosin. But maybe you have muscle that is in your intestines, or muscle that is in your heart, or muscle that is in other parts of your body. And so with just one gene, you can get different kinds of proteins. And that's similar to like computer code. With computer code, you kind of want your information to be as brief as you can possibly make it. And that's kind of the same way that it works inside of these guys. Okay. So that is splicing. Next, we've got this 5' cap and this thing that is called a poly A tail. And so the 5' cap means that we have got, okay, so 5' cap. That means that we take our piece of messenger RNA and we are going to add bases to the 5' end. And those bases are necessary. They allow the messenger RNA to leave the nucleus, because the nucleus is a really high security, really special sort of a place that only certain things are allowed in and certain things are allowed out. And so the 5' cap is put on the 5' end of the piece of messenger RNA, and when it's there, it is going to be like the little boarding pass so that it can leave the nucleus. The final piece we're going to get is a poly A tail. And that is because your body, the second that it sees any nucleic acids in the cytoplasm, will immediately digest them. And that happens with all organisms, with all sorts of cells, which is that all nucleic acids in the cytoplasm, so that means that if it's whether it's RNA or DNA, will be destroyed. And they're destroyed by digestive enzymes. It's kind of like an immune system within your cell. And so that seems kind of stupid. That means it's going to digest information that was intended to be there in the cytoplasm, namely your own messenger RNA. But this is actually an easier sort of defense than having a whole other system to determine what should be there and what shouldn't. And so your body just digests any RNA or any DNA that it ever finds inside of the cytoplasm. So that means that the second that this messenger RNA arrives into the cytoplasm, it is going to start being kind of eaten apart by enzymes, which are going to be chomping away on that thing. And they're doing that because they think this might be an invader. And those enzymes, they read or digest the messenger RNA in the same direction that everything else has, which is always from 3' to 5'. So the poly A tail is on the 3' end of the messenger RNA. And its job is to basically be a fuse, if you want to think about it that way. While the poly A tail is being digested, you're firing off proteins off of this transcript the whole time. So eventually this enzyme is going to catch up and it's going to actually destroy your little piece of messenger RNA. But by the time that happens, you've hopefully fired off a few copies of the protein. And if you need more of that protein and this thing is gone, that's okay, because you're going to keep popping out more and more copies of messenger RNA. And then when you don't need that protein anymore, you just stop making messenger RNA, the enzymes eat up all the transcripts, and the signal just goes away. And so it's kind of a self-regulating process. And so if we go back to what we started with, we start with our messenger RNA. And when we start with our messenger RNA, it's going to have a mixture of exons, introns, After splicing, we're going to have nothing but our exons inside of there, because remember, those are the ones we want to keep. After we add our 5' cap, we're going to have our exons, and now we have our 5' cap. And then by the time we do the last step, we're now going to have exon. Here's our 5' cap, and then on the other side, we've got our poly A tail. And by poly A, we just really do mean a bunch of A's. So that base adenine, A's, 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 we just put a whole bunch of A's that don't code for anything, but they're going to over here be consumed as soon as they go into the cytoplasm by these enzymes. But by the time that that is chewed apart, we would have already fired off a couple of proteins, and that's going to allow the entire process to work.